So I'd like to talk about some discoveries we've made just by applying nanopore sequencing directly to sequencing phage genomes from phage collected in the environment. So we can ask a couple basic questions first. Why study viruses and how do we study them? And I took this really nice diagram from a paper that Asaf Vardi and his colleague wrote recently in PLOS Bio, and I like it because it sort of shows it's complicated. So we study the ocean in many different ways, from satellites looking at phytoplankton blooms from space to sampling in situ. And one of the reasons why viruses are an element of study in the oceans is because they can affect how carbon from the surface of the ocean either makes it to the deep sea and gets sequestered or doesn't. And they do so in a variety of ways. If you look at sort of in the center right of this slide, you'll see this thing says viral shunt. Well, phytoplankton fix CO2 from the atmosphere. And one thing that viruses can do is infect those algae and then lyse them, and then that carbon gets re-released. So it never makes it down into the deep sea. But other viruses actually, when they infect cells, can cause aggregation. When that aggregation happens, that carbon can make it down to the deep sea, where it's sequestered from the atmosphere for hundreds of years potentially. So oddly enough, thinking about the very big, that is CO2 in the atmosphere, which we contribute to, is influenced by the very small nanometer-sized viruses. And these are some of the driving motivators for doing this research. This is just repeating a bit of what I said, but it's important to understand that the ocean stores maybe 25% of anthropogenically produced carbon. And viruses are abundant, by the way. So in a milliliter of seawater, you might have 1 million microbes. There's 10 times more than that of viruses, so maybe 10 to the 7th viruses. So there's a lot of them. They can either release that carbon at the surface by lysing the cells they infect, or sometimes they can cause this aggregation, which causes sinking. So folks in oceanography are still trying to sort this out in terms of the influence of viral biology on the carbon cycle in the ocean. And so those are some of our main drivers. These little fellows you see here are actually from some of our concentrated samples, individual bacteriophage that we've sequenced genomes of using the approach. Now, the challenge, of course, in trying to get viral genomes using shotgun metagenomic approaches is, as we've heard before and we'll hear for a while, is assembly. And this is from a paper of one of my former postdocs, and they quite well state that most viral contigs you generate from shotgun assemblies from complex communities are highly fragmented. And this, of course, poses challenges to downstream analysis and even understanding the structure of viral genomes in situ. So what we did initially came from a collaboration with our colleagues in the New York office of Nanopore, Cecil Jewell and Matt Pendleton, and then later on with John Bollier, who's in the San Francisco office. So this was our launch into sequencing viral genomes. And what we were able to show is that when we basically concentrated phage from seawater and then extracted DNA from that and sequenced it directly, we got full phage genomes in individual reads, which was pretty exciting for us. And in fact, you could look at the gridiron traces coming off, look at the sequencing, and because capsids basically protect the DNA, we could basically see the size of the viral genomes because they tend to be in particular categories. They're quantized, if you will, 30 kb, 40 kb, 60 kb, 80 kb. You can actually see that coming out in the nanopore traces. And this is just an example of the colored at the bottom left there. You see basically the sizes of genomes from different depths in ocean waters out where we work, north of Hawaii, from the surface or near surface 25 meters, 117 meters, and 250 meters. So we get intact viral genomes, and John Bollier established this first workflow where we could identify those phage and then start to sequence them. But some of the sequences we got, which were the size of a phage genome, turns out they weren't phage genomes. And that's what really is this talk is about. So one of the things that we discovered in this analysis, and if you look kind of in the center of the slide, the blue dots in that one plot, those basically, these are genomes that are, say, 40 kb or 60 kb, but they weren't a full phage genome. Rather, they were a series of repeats that ranged from maybe 5 kb to 8 kb, but they always equaled the average size of these genomes in situ. So 5 times 8 is 40. That's one of those little dots there, the clusters of the different genomes we found of these concatemers of monomeric elements and concatemers that equal the size of a phage genome. And over to the right of that slide, you will see some of those concatemers. These are about 40 kb in length, many of them. And those yellow genes identified, actually, those are all basically integrases, which gave us our first clue about what these concatemeric elements might be. And so what we decided to do was to extend this workflow, actually work a bit at the front end, so we got highly purified phage preps, and then see what we could find. But before I go further, I can tell you we had a hypothesis going into it when we saw those integrases, and we thought, well, these might be something that are known as phage satellites. So phage satellites are mobile genetic elements. They reside in the genome. They can be anywhere from, say, 5 kb to 15 kb. But when a helper phage infects a cell, call them helper phage, they basically will pop out of the genome, circularize, and then make their own genomes, and also trick that helper phage into packaging their DNA in capsids instead of the host DNA. They have a variety of tricks for doing that I don't have time to talk about right now. But we postulated that that was going on. This is from Kimberly Seed's lab, this figure in the middle with all those bubbles in it. And you can see that there's this phage infecting. This is from Vibrio cholera, actually. And when there's an element, that PLE that you see in blue there, when it senses that there's a phage infection, the ICP phage, then it pops out and it fools that phage into packaging it instead of the original phage genome, and then propagating in that manner. Now, our discovery in this work is that some of these phage satellites actually appear to make concatemers, which are then packaged by the headful into these phage genomes. So, into these phage capsids, and that's how they propagate. So what we did was to refine our approach, and we basically were able to get highly purified phage preps. And the data I'm going to talk about right now comes from just one seawater sample. And what we were able to do was to sequence it. We've got a lot of phage genomes that I don't have time to talk about right now. But we also found 2,000 new of these phage satellite elements from one seawater sample. And this is our workflow, and basically after purifying the phage, we sequence them, we get full-length phage genomes as well as full-length concatemers of these phage parasites. And we were able to take then the monomers, map them back onto genomes to see what genomes they reside in. And in fact, these particular phage satellites fell into some of the most common bacteria that reside in seawater, and so they appear to be pretty common. I don't have time to talk about the phage, but this is an example of alignments of some of the pelagiophages that we found out in these environments. Let's talk a little bit more about the parasites since I'm running myself out of time. So we were able to cluster them, and everything you see in those blue clusters are these new phage satellites that we found. The known phage satellites on the outside are in colors, and those correspond to phage-inducible chromosomal elements, Peachy-like elements. And these are not inconsequential, by the way, because the toxic shock protein, how that moves around, is on these phage satellites. So they have significance. Some of the major genes that they have are shown in those boxes on the side. Those are shown basically in order of appearance, or the most abundant ones throughout the genome. There's phage integrases, there's primases, ways that move these things around, and also some cargo, if you will. So they move genes around as well. We can look at the phylogeny of these things. Don't have time to talk about it too much, but all those things in blue there are unique phage satellite elements from these populations of bacteria in the sea. So these things basically can hijack phage within these cells and propagate in this manner. So these satellites are way more abundant than we thought. And this is just a heat map showing we see them both in cells as a function of depth and over time very commonly, and they're pretty abundant. So to wrap up, these are new pirates of the sea. They're basically the parasites of the parasites, and they're abundant. Back of the envelope calculations, there's probably minimally 3 times 10 to the 26th of these things out in the ocean. They have a lot of diversity and probably a lot of different mechanisms for hijacking their helper phage. And so I think any place where you find bacteriophage, you're going to find these things. We never would have seen them with short-read sequencing because we never would have found the concatemers. And so this is a great example of where nanopore sequencing really opened up a new window into a biological entity that we really didn't know existed before we applied this tech. So I think I have to leave it at that. I want to thank John Epley, who helped lead the charge on this study, Andy Berger as well, other folks in my lab, Steve Biller at Wellesley, who helped us in phage purification, and our colleagues at ONT, John Bollier in San Francisco, Cecil Jewell, Matt Pendleton in New York City. Thanks for listening.